大学堂顶尖学者讲座啊，这我还得介绍几句，因为这是必须的啊。是2012年北京大学为了推进这个双一流的这个发展决定每年邀请接近10位顶尖学者来北大讲座。那么张峰呢是第92位，每年我看现在还不到10位啊。那下面这个我就用英文说了啊。I'm honored to introduce and welcome today's speaker, Professor Feng Zhang. Feng is the core member of the Broad Institute and investigator of McGovern Institute for Brain Science. James and Patricia Hoyt, professor of neuroscience at MIT. 你看他有多少抬头？Now this is a big one. Carl Hughes, investigator. Carl Hughes, medical investigator. He is the member of the National Academy of Sciences, the American Academy of Arts and Sciences, as well as a fellow in the National Academy of Inventors. Feng is a molecular biologist focused on improving human health with amazing track record. Okay? He was born in Hebei. He went to undergraduate study at Harvard, where he worked in the lab of Zhang Xiaowei, just down the hall from my lab. So we knew each other from that time. I was most impressed with Feng being an undergraduate. Okay, now in our labs we all have undergraduate researchers. He was only second year, third year undergraduate. He was spearheading the biological research in Xiaowei's lab. Xiaowei, many of you know, was trained as a physicist, just arrived at Harvard, learning biology like me, right? He knew physics, he also knew biology. And so what happened from that point on, I was not surprised why he accomplished so much at such a young age. He then went to Stanford. Actually, we tried to convince him to stay at Harvard. I wasn't sure I should talk about this. At the time he left, he came to say goodbye to me. And he left me a cute stone, a bird sculpture. I still kept it at my home in New York City, where my daughter lives. So he went to Stanford and joined the laboratory of Carl Hessel, where he collaborated with Ed Boyden and invented the optogenetic technique that was widely used worldwide. It's very important brain research tool. That was his graduate work. And then he started at Harvard as a junior fellow. This is when he got interested in gene editing. I think from that point on, he probably knew what he did. He was the first to apply the CRISPR technique to mammalian cells that enabled the mental applications of gene editing techniques. He accomplished a lot. He has many companies and trained many students. And I think I should just stop here. And the title is exploration of biological diversity. Please join me to welcome Feng.
大家好。我们一起拍一个自照，好不好？ OK， 好。真是非常感谢大家的热情，我觉得很激动来北大，因为我是在石家庄出生的，然后在河北长大，一直上完小学大家可以听见我说话吗 ？OK， 那我就不用这个了。那个我是一直就是长到11岁，然后去了美国，但是在中国小时候上学的时候，别人总是说北大是中国最好的大学。所以从小的话，我就一直有这个印象，就是，要努力，要争取学习好，然后可能你的运气会足够好，然后你可以去上北大去上学。但是当时上学的时候，我也知道，其实我要是在学习的话，然后考试，其实我考到北大的可能性是很低的，因为真是有很多聪明人，而且很多聪明而且努力的人，所以呢，真是今天能来到北大来给你们讲一讲我做的事情，真是非常大的荣誉。而且呢，我真是非常激动，可以来这里和大家来讨论一下，然后来就是 talk science 所以非常 exciting。OK， 好，那我是11岁从石家庄搬到美国去，然后在那边长大的，然后就一直就是对科学很感兴趣，然后我觉得我一开始对生物没有特别大的兴趣，因为我觉得生物，因为上初中时候上生物课，老师给你一个非常就是难闻的一个青蛙，然后让你把它做一个那个dissection， 然后你再背很多各式各样的乱七八糟的东西，然后就是觉得特别枯燥。但是我上7年级的时候，我去了一个就是周末的一个小课程，他教 molecular biology， 然后上那课时候，他们给我们看了一个 movie 叫 Jurassic Park， 然后就是边看这个 movie， 然后边听老师讲 DNA、RNA、central dogma 这些东西，然后就让我觉得可能这 movie 里讲的做 dinosaur 这些东西，其实可能是可以实现的。然后我就觉得这个挺有意思，然后就慢慢就开始对生物越来越感兴趣。然后我在高中时候，当时有一个机会去我的城市里的一个医院里，他们开了一个新的这个基因治疗的那个研究所，然后他们收 intern 收 volunteer， 然后我就开始去那儿去工作，然后就慢慢的就开始学习一些 molecular biology 的 experiment。当时GFP 刚被发现，然后我就是教我的那个 scientist， 他教我怎么把 GFP 的基因放到一个细胞里，然后带我到一个非常黑暗的一个显微镜的房间里，然后我把细胞放到显微镜上，然后一看，哇，所有的细胞都是绿的，从来没有看过，真是从来没有看过那么漂亮的一个东西，所以当时我就觉得这个就是这种 science 真是非常酷，非常有意思，所以就从那里开始一直在学 biology， 然后上大学的时候从三立那儿学 biophysics， 然后学怎么着用 quantitative thinking， 然后来学习 biology， 然后还学了很多其他乱七八糟的东西，反正就是从那里开始的。但今天呢，我就跟 我想给大家讲一讲我最近做的一些科学。我的中文就是小学5年级水平，所以呢，我的 talk 我就用英文讲了。Okay. So you know, I've been really inspired by genetic medicine, and genetic medicine is exciting because it's modular. It provides a new way to think about how to develop therapeutics for treating disease. So I put over here this picture. It's a rocket sending a satellite into outer space. And this modular concept applies to genetic medicine as well. So really there are two pieces. There's a vehicle, so the delivery vehicle that can send the therapeutic molecules to the right cells, to the right organs in the body. And then there's also the payload, which can go into the cell and do whatever you want. It could be mRNA, could be gene editing, could be epigenetic regulation, whatever you want to do. So the idea is if we can develop a suite of vehicles that can go to the brain, go to the liver, go to the kidney, and combine that with a suite of payloads, then we can really have genetic medicine to treat many, many different diseases. This payload part has advanced quite a bit. There are a lot of different payloads that we can use now. But the vehicle technology has lagged behind. So if you look at clinical trials today, the most successful ones target 3 tissues: liver, eye, and also the blood. And the vast majority of other tissues, we don't really have a way to target them. If you want to put something into the pancreas or into the kidney, there are a lot of diseases, and we have strategies to treat them, but we just don't have a good way to get the molecules into those cells. So for today, I thought I'll share 3 things that we've been working on. I think they're kind of interesting. One of them is on human capsid-forming proteins and harnessing them for delivering things into cells. And then a corollary of that is the second story, where prokaryotes, bacteria, have also acquired viral components, and we can harness them to be able to deliver into cells. And then lastly, just a brief description of some of the things we've been doing on looking at CRISPR-related systems and harnessing them for RNA-guided DNA targeting and DNA modification. Okay, so we'll start with the first one. So, we encounter a lot of viruses, and so there are many viruses. There are DNA viruses, there are RNA viruses, and today I want to talk about RNA viruses. And so there are 2 types of RNA viruses. There are RNA viruses like corona or influenza. They have an RNA genome, and they replicate in RNA form. And so what that means is if your body clears the virus infection, all the viruses disappear. The second type of RNA virus are retroviruses. HIV is one version of retrovirus. They are different. They have an RNA genome, but they replicate via reverse transcription. So the RNA gets converted to DNA, the DNA integrates in the whole cell's genome, and then it makes more virus from there. So over the course of evolution, hundreds of millions of years, our evolutionary ancestors all along have been bombarded by retroviruses. And so in fact, if you look into the human genome, 40% of our genome are retroelements. So they all came from accumulation of these past viral infections. So needless to say, there are a lot of viral things, retroviral things in our genome. And so when the human genome was first sequenced more than 10 years ago, scientists began to use bioinformatics to analyze the sequences in the human genome. So there are endogenous human retroviruses, but then there are also human genes. And so ignore the human endogenous retroviruses. And this paper from Campillos and colleagues, they looked at the 20,000 or so protein coding genes in the human genome. And they found that about 80 or so out of the 20,000 are retroviral-like. And so here are some of them. And what you see is that these genes, they have this blue domain, which is highly homologous to the retroviral capsid domain. Capsid domains form the virus structure, and then they package the RNA, they package enzymes in this capsid and protect it. And so what they've found is that there are a lot of human genes that are derived from this capsid-forming part of the retrovirus. And they group them into different groups. There's the sushi family. It's called sushi family because they came from the sushi retrotransposon. So that's the evolutionary lineage. And then there's the scan family, the paraneoplastic family, and then there's also this gene called ARC, which is expressed in the brain. So what got me really excited was that almost 10 years ago, there were two studies, one from this Pastusan, Jason Shepard's lab at the University of Utah in America, and then also Travis Thompson's group, Ashley and colleagues at University of Massachusetts, also in America. They were studying the ARC gene. Jason was looking at ARC gene in mice, and then Travis was looking at ARC gene in Drosophila. And what they found is that this ARC gene is expressed in neurons, is turned on, and is released by neurons in the form of these virus-like structures. So that was really exciting. That was the first demonstration that these human endogenous GAG-like proteins are able to form capsids and are released by cells. Even more interesting is that in Drosophila, this capsid protein packages the mRNA encoding ARC itself. And moreover, is secreted by neurons and is sent into the downstream postsynaptic muscle cells. So it works at the neuromuscular junction. So that was very, very interesting. And we thought if ARC is one of these proteins and it can potentially mediate intercellular transfer, maybe we can study them and harness this broad family of proteins to be able to package therapeutic molecules and be able to deliver to downstream cells. So Mikey and Blake are two of my lab members, and they embarked on a study of these endogenous GAG-like proteins. And so here are 5 different proteins that they studied. What they did is that they put the gene, the human genes, into E. coli and purified synthetic protein. So recombinant proteins out of E. coli. And what they found is that 4 out of these 5 genes, so these 4, they were able to, maybe I'll use a cursor. So these 4 genes, they are able to spontaneously form virus-like capsids. And so that was really, really interesting. So this showed that there is this conserved property of all of these human endogenous GAG proteins. They mostly preserve their ability to form capsid structures. And so the next thing they did is that they put these different genes into human HEK293 cells. And they looked to see, like ARC, are they also secreted by the mammalian cell? So what they found is that at least 4 of these proteins are secreted by HEK cells, and one of them, called PEG10, is secreted most abundantly. And it's always easier to study things that are very abundant, and so we decided to focus on studying PEG10. The next thing we did is we wanted to see whether PEG10 is similar to ARC. Does it also package RNA, and does it package its own mRNA? And so in this study here, we extracted RNA from these PEG10 particles that are secreted by HEK cells, and we extracted RNA, and we did RNA-seq, and we found that is indeed PEG10, the mRNA is enriched in these PEG10 particles. So that was kind of nice to see. It confirmed the property that was also observed for ARC. So then we try to figure out how is this RNA getting loaded into the PEG10 particle. And we found that there are a couple of things. There is a signal on the 5' UTR of the ARC mRNA, and then there's also a signal on the 3' UTR. And if you replace the sequence in between, so if you get rid of the ARC mRNA, the ARC encoding gene, and replace it with a new RNA, that new piece of RNA, this synthetic RNA, can also get loaded into the PEG10 particle. And so this allowed us to then develop this 3-component system. So if we have a plasmid that expresses the PEG10 protein, a plasmid expressing the synthetic RNA with the packaging signals, and a fusogen protein, altogether they can be used to be able to produce PEG10 particles. And so indeed, if you look on the right side, you see that these are EM images of the particles that are secreted by cells, and they look like retroviruses. And this negative control shows what exosomes look like. And so you can see they are very different looking. And this is just to show that we can make these synthetic particles. So then we can load the Cas9 mRNA and also guide RNA into these particles. And then we can use it to deliver mRNA into target cells. And so, so without too much optimization, you can get a reasonable amount of gene editing in these experiments. So we were really excited at this point. We thought, okay, so we can make these particles. Maybe we can make more of them and start to do mouse studies and see how well these endogenous particles fare as gene delivery systems. So science goes up, and oftentimes it comes down. And so this is exactly what happened here. So as we were getting excited to do a mouse study, we thought, okay, let's make enough particles. So I remember one day at lab meeting, so we have lab meetings once a week, and Mikey and Blake were presenting, and they showed a slide. And at first, I didn't understand what the slide was talking about. So they showed a stack of Petri dishes, 15-centimeter Petri dishes, stacked from the surface of Earth all the way to the geosynchronous orbit. And so it turned out that to do this experiment here, it was a heroic effort. For a single well on a 96-well plate, about 5,000 cells, they had to grow a single 15-centimeter plate and concentrate everything down into 20 microliters to then put onto the single well. And so we were trying to do mouse experiments, right? So we're trying to target hepatocytes in the liver, and there are tens of billions of cells there. So it turned out that in order to make enough particles to do a proper mouse cohort, maybe 5 to 10 mice in this experiment, they had to grow enough plates such that if you stacked it up, it would go from the surface of planet Earth all the way to above the geosynchronous orbit. Very clear this wasn't going to work. And so this is when science became quite depressing. But anyway, that's okay, because you try multiple things and sometimes science can come back up. And so at the same time, Vicky and Yugang and Ramya, 3 other lab members, they were studying a different subset of these endogenous human proteins. And that is the PNMA family. And we were using a similar strategy. We identify the genes, we put them into human cells, we look for secretion, and we characterize packaging. So we took all these different PNMA genes and put it into HEK cells. And interestingly, they found that one of these proteins, called PNMA2, is secreted from cells as a protein-only capsid. So PEC10 is secreted as a membrane-bound particle, so it's just like a lentivirus or retrovirus, has a lipid envelope. PEC10, on the other hand, is like AAV. It gets secreted as a protein-only capsid. And so this is what they look like under EM. These are the protein-only capsids. That was potentially very interesting because if we have a capsid that doesn't require a lipid membrane, it makes it potentially feasible to make these particles fully synthetically, fully recombinantly out of sort of bacterial purified protein. And if we can do that, then we can potentially scale this much more. And so, so then we try to characterize PNMA2 more to understand packaging. And sadly, we found that it didn't package any mRNA. So over here on the right side is the same experiment where we took the supernatant, extracted RNA, sequenced it, and we found there's no enrichment of any RNA in these sort of extracted fraction. But that's okay. We thought, let's understand PNMA2 more. Maybe we can figure out how to package things into it. And so then we continue with the biochemistry experiment. And what we found is that the PEC10 protein expresses very well recombinantly. So if you use E. coli to express it, you get very, very nice protein yield. And the protein spontaneously form these capsid structures. And they are a well-behaved biochemical system. So if you drop the salt concentration to 0, the capsid will fall apart. And then if you add salt back in, the capsids will reform. So now you have a very nice cage where the cage will form, you can open it up, and then you can reform it. And so that makes it possible to potentially package things into it. But we also had to figure out why is it not packaging RNA by default. So we solved the cryo-EM structure. And so this is a cross-section of the virus-like particle. The color shows electrostatics. So red means negative charge, and blue means positive charge. And you can see that inside of this capsid is very negative. RNA is negative, nucleic acids are negatively charged. And so it's not surprising that there's no RNA or nucleic acid inside of these cages. But this gave us an idea. Maybe we can go and engineer the surface charge and change it to positive so that we can get RNA loaded. So we took the wild type PNMA2 protein. We removed a disordered, highly negatively charged peptide at the C terminus. And we replaced it with a well-known RNA binding domain from the CCMV virus. And this is highly positively charged, so you can make this engineered PNMA2 protein. So this engineered PNMA2 can continue to form very nice-looking virus particles, and just like the wild type PNMA2. And so it's a very nice system. And then we found that by making this simple change, we can now package RNA into these particles. So if you take sort of opened up single monomeric capsid proteins and you mix it with RNA, and if you add salt back in, they will form these capsids with RNA inside. So then this allowed us to start to do some delivery experiments. And still early days, and we're still working on it. But in this experiment, right in the middle column here, we took the engineered PMA2 protein, and we used it to package the Cree mRNA. And then at the same time, we treated the particles with RNAs so that we're getting rid of everything that's on the outside of this capsid. And then by putting it onto target cells, this is a reporter cell line that has the Flox GFP. We can get mRNA for Cree delivered inside, translated, and then it can activate expression of the reporter protein. So the expression level is still modest, and I think that is due to a couple of different challenges. Number one, these particles probably don't bind to the target cell all that efficiently, so we have to improve targeting and binding. And then second, once they bind and are endocytosed into the cells, they probably don't come out of the endosome as efficiently as we would like. And so the release is also something that we need to improve. But nevertheless, there's some signal, and a signal provides a way to be able to further optimize and to make the system better. And so we're working on that now. So hopefully next time I'll be able to tell you about some of the progress that we made on that front. Okay, so that's the first story. So keep in mind, so the challenge that we had with these particles is that the release efficiency is not efficient. And in fact, endosomal escape is a major problem for any type of molecular delivery. Probably many of us here have had the coronavirus mRNA vaccine. And so the mRNA vaccine is a lipid nanoparticle-based delivery system. So the dose that we receive is usually 100 times more than the amount of mRNA that is actually needed to express enough of the epitope. And that is because the endosomal escape efficiency of a lipid nanoparticle is 1%. So very low. So we get 100-fold more stuff injected into our body. So we've been thinking about how do we make this escape efficiency higher. And so Joe Kreis here has been thinking about using an injection system from prokaryotes to be able to deliver things directly into the cell across the cytoplasmic membrane and bypassing the endosomal process. Because if you can do that, then whatever you are injecting to the cell, that is directly already accessible to the machineries in the cell. So to do this, we were interested in bacterial secretion systems. So bacteria have evolved many different versions of secretion systems that are usually used to be able to secrete effector proteins either to other bacteria or plant host or eukaryotic host. But most of these systems are bound in the bacterial membrane. And so if you wanted to use these systems, you have to inject bacteria into the host organism. And that is sometimes not ideal. So we thought, can we find a secreted form of these injection systems? After all, there are natural examples. Phage viruses, they're naturally secreted injection systems. So it turned out that there is one particular system, a Photorhabdis virulence cassette, that is secreted. And so actually really amazing work has been done here to solve the EM structure for these PVCs. And so what these things are, they are secreted phage tail-like systems that can use a spring-loaded mechanism to inject proteins into target cells. So here on the left side, this is a kind of a schematic that shows how this may work. You have a spring-loaded system that has a payload loaded inside. They have these tail fibers, these gray kind of leg-like structures. They can land onto a target cell, recognize, and then engage the spring-loaded mechanism to then push a very sharp spike protein across the cell membrane. So this is the spike protein. So once this thing lands on the cell, it will push this through the cell membrane and then deliver a payload into the target cell. So we thought this is really cool. Maybe we can somehow understand and then co-opt the system to be able to deliver proteins into target cells. So the way that this system naturally functions is that they live in a pathogenic bacteria called Photorhabdus. And this bacteria will secrete these they're called contractile injection systems, or CSs, but we'll just call them syringes. They will secrete these syringes, and the syringe will go and bind the intestinal cells in this insect and lyse off the intestinal cells. And this will kill the insect. When that happens, the bacteria can then escape the insect and colonize more insects. And it's just part of the natural life cycle of this bacteria. And if you go into the genome of this bacteria, you will find all of the molecular components. So here on the left side is a operon that encodes all of the structural proteins, everything that makes up this entire syringe structure. And then on the right are the payload genes. And these are usually toxins. And they all have a signal peptide on the front of this gene. And that's how the protein gets loaded into these syringes. So if you have all of these different components in a bacteria, you can make new syringes. And so we thought, okay, let's work with this. Can we transfer everything into an E. coli and be able to mass produce these particles? So that worked. So here in the middle is an EM image showing what these E. coli produced syringes look like. You can see they are long and narrow, and they actually have some variable distribution of length. Some are quite long. This is a really long one, but most of them are about the same length. So there's this kind of a distribution. The longer ones can package more molecules. The shorter ones package fewer. They just don't have as much loading capacity. But nevertheless, this shows that the system can make new syringes in E. coli. So then we wanted to see, are these recombinant syringes functional? And so we just packaged toxins into them, and then we try to see, can they target and inject the toxin into insect cells? So here we use SF9. It's an insect cell line. And what we found is that if you have the wild type system, so fully reconstituted syringes with toxin loaded inside, it will kill insect cells pretty effectively. And as negative controls, you can delete the toxin from the inside, or you can get rid of the binding tail fiber, and all of those abolish the killing activity. So this is a good sign. It shows that we can make functional syringes using E. coli. But what we really wanted to do is to use it to target human cells. And so Joe tried a lot of different things. So the basic idea is that we wanted to engineer these tail fibers by replacing the insect binding domain with something that binds to human cells. And he probably tried more than 100 different designs without knowing what the structure looks like, and all those failed. And so we were about to give up. Because you try it 100 times and it doesn't work, it's not going to work. But at that moment, AlphaFold2 came out. So you see where this is going. And we thought, let's give it one more try. So we put the sequence for the tail fiber into AlphaFold2, and we got this really interesting structure. And so based on that, Joe thought, okay, let's cut it off right here. Maybe this is the binding domain. And then we can then fuse something else to it. So we tried either the human adenovirus 5 binding domain, or we tried a DARPin that can bind to EGFR. And just as negative controls, to make sure that we're seeing function because of effective binding onto human cells, we made a mutant Ad5 binding domain that doesn't bind. And then also we used a DARPin that recognizes a lysozyme rather than EGFR. So these are just controls. And then we used a reporter cell line, this A549. Is AD5 targetable, is also EGFR positive. It's a GFP reporter cell line. And so now we loaded the Cre protein. We can put a single peptide on the N-terminus of Cre so that protein gets loaded inside. And we thought to see, can we activate the reporter gene? This is a wild-type tail fiber, no signal. And of course, if you remove the binding domain, there's also no signal. But then once you put a human binding domain onto these tail fibers, you can get successful activation of reporter. So AD5 gives you signal. If you inactivate the binding, it doesn't give you signal anymore. EGFR binding also activates reporter, but then lysozyme doesn't activate reporter expression. So this is really exciting. It showed that we can retarget this to human cells. And then we switched out Cre. There's no Creitis, so we wanted to treat something that's more relevant. So we put Cas9 and also a zinc finger deaminase to be able to do base editing. And so what we found is that we can get reasonable levels of signal in the target cells. And then we wanted to further understand how modular this binding or this retargeting strategy is. And so we made different tail fibers. Here across the columns are different tail fibers engineered with different protein tags. So HA, flag, EE, etc. And then we also made reporter cell lines that express antibodies that are capable of binding to these different tags. So we have cells that can, reporter cells that are anti-HA, or reporter cells that are anti-flag, and so forth. And this was really nice to see. Here, what we found is that only when the tag on the syringe matches the antibody on the cell surface in their binding, do you get reasonable activation signal. There's some low levels of background, but by large, you only get successful targeting and reporter activation when the peptide and the antibody are a match. And so this shows that this is potentially a nice reprogrammable system. But in order to make this work, you really have to have a unique marker on the cell surface, and you also have to have a good antibody that can bind to that marker on the cell surface. But anyway, so then finally, another thing we tried is we injected it into the mouse brain. We wanted to see, can it target cells in vivo, and also does it work? So we have a reporter mouse that has tandem dimer tomato flocks so that we can activate its expression. And then what we found is that if you stereotactically inject into the hippocampus, you can get expression in the hippocampus. The distribution, the diffusion is limited, I think because the syringes are large, they're long, they're usually 100 nanometers or even longer for some of them. So distribution is not as good. So we're still trying to figure out better ways to get them to distribute better. Maybe there are active transport processes that we can take advantage of, but something that we haven't yet fully figured out. But nevertheless, it can work in vivo. So we're also excited about this approach. But I think basically there are a lot of ways that nature is able to shuttle molecules back and forth between cells. And exploring some of these mechanisms can potentially help us develop new delivery systems. And so that's kind of the general strategy that we're taking. Okay, last but not least, just a quick update on some new RNA programmable systems. So as you probably know, gene editing has really been made possible because of these DNA binding domains that you can reprogram. Be it zinc finger proteins or transcription activator like effector proteins or Cas9, the thing that made it work is the ability to reprogram it to recognize a new DNA sequence. And so over the years, over the decades, the progression in these gene editing systems is really the dramatic simplification of DNA targeting programmability. Meganucleases are really hard to reprogram. Zinc fingers made it easier, talons easier, And then finally, CRISPR-Cas9 just made it really easy to reprogram because you just synthesize a new piece of guide RNA. And so because of that, we and also many others have been very interested in RNA-guided DNA targeting systems. And so CRISPR is one of the places where there are a lot of these different RNA-guided systems. And so we focus on looking at CRISPR diversity. There are two major classes of CRISPRs. There are class 1 and class 2. And we mostly focus on class 2 because they're simpler. They use a single protein working with a non-coding guide RNA to bind it and to achieve effect on the target cell. So Cas9 was the first class 2 CRISPR system that was really well characterized and developed for gene editing. But there are many more. There are also the Cas12 family of RNA-guided DNA endonucleases. And there are also RNA targeting systems too. So Cas13 systems target RNA because there are RNA viruses in nature in addition to the DNA phages. So bacteria have also evolved that. And so all of these different things have together formed this ever-expanding CRISPR toolbox. You can either use the nuclease function for gene knockouts or to stimulate homologous recombination, or you can use this system as an RNA-guided DNA binding domain to then be able to recruit different effectors, whether it's for gene modulation or base editing, prime editing, etc. You can do a lot of things with it. But one of the challenges is that these systems are generally large. Cas9 is 1,300 amino acids long. It's a large molecule. So it's hard to deliver sometimes. And so Han and Sumia are two lab members that embarked on a bioinformatics study of the evolutionary path of Cas9. Because Cas9 is really large, and we wondered, did it come from something that's simpler? And so by taking some of the key hallmarks of Cas9, the nuclease domains, and searching throughout all of the microbial sequences, they were able to reconstruct the evolutionary history of Cas9. And so over here, this big circle is the evolutionary history of Cas9. And what they found is that Cas9, or what we call Cas9, is one very specific emergence out of this evolutionary tree. And they came out of a broader family of proteins called ISCB. So ISCB is a transposable element, or is a protein associated with transposable elements. It's very abundant. Sometimes in a single bacterial genome, you can find 300 copies of ISCB. So they're as abundant as ribosomal genes. And ISCB is very diverse, and so there are many different major evolutionary paths that it has taken over history. And over the course of time, some ISCBs have even acquired association with CRISPR-like repeats. And over here, whenever you see these blue bars, these indicate association with CRISPR. And so Cas9 is this really successful emergence of association with CRISPR. So a lot of them are CRISPR associated. But then occasionally there were ISCBs that also acquired CRISPR. They didn't quite become Cas9, probably because they weren't robust enough or they didn't serve sufficient selective sort of evolutionary function. But nevertheless, they came to be. I guess another way to think about it is if you think about human evolution, there were many different versions of human. We are Homo sapiens, but there are also other homos, Homo Neanderthal, Homo denisovans, so forth. And so Cas9 is kind of like Homo sapiens. And then these other ISCBs are the other homos that somehow did not sort of succeed in terms of evolution. So what is really interesting about ISCB, and even this even earlier ancestral form called ISRB, is that they are compact. They are about 25 to 30% of the size of Cas9, so very, very small. And if you look at their protein domain organization, they are basically the most minimum sufficient form of Cas9. They have the nuclease domains, but they don't have this very large rect domain. We know Cas9 works with non-coding RNAs to achieve DNA targeting and also DNA cleavage. It wasn't clear whether or not ISCBs also had a non-coding RNA. But keep in mind, we saw these occasional emergences of IACB with CRISPR association, and so that gave us a hypothesis that there's probably a non-coding RNA working with IACB. And so to test that, we then did RNA sequencing. So here we did an RNA sequencing experiment where we look at the upstream region of IACB. And there we found a non-coding RNA, a small a medium-sized non-coding RNA, sometimes 300 to 500 base pairs long. But that is quite robust. And if you look at the secondary structure, it's very well formed, and that told us that maybe this is a non-coding guide RNA. So this is how the whole system came about. We studied that, and it turned out that IACB indeed is the ancestral form of Cas9. It has the same mechanism as Cas9. It uses the non-coding guide RNA and two different nuclease domains to independently cleave the two different strands of DNA. So everything kind of checked out. And structurally, IACB and Cas9 share similarities in the core nuclease domains. So here, in this kind of yellow-orangish color is the protein. Gray is the scaffold of the non-coding RNA. Blue is the DNA that's being targeted. And then this red fragment is part of the non-coding guide RNA that binds to the DNA. And then over here is the same color scheme. So what you see is that Cas9 is very small in terms of the RNA scaffold. IACB is very large, very, very elaborate. But Cas9 has much more protein domains, and IACB doesn't. So basically what happened over evolution is that the RNA part that is used to stabilize this RNA DNA heteroduplex, this R loop, has been gradually replaced with protein domains. And so this is what you see in the case of Cas9. And so this is basically what happened when proteins needed to become more and more robust. Proteins are more robust than RNA. And so in order for IACB to become a bona fide immune system in bacteria, it acquired all these protein components so that it's robust, stable, and can really kill bacterial viruses once they enter into the cell. So that's how Cas9 came to be. But nevertheless, IACB is small, and that compactness provides some potential utility for therapeutic or gene delivery applications. In addition to IACB, we also found the ancestral form of Cas12, and that's the TMPB protein. But they all work kind of in the same way. And then last but not least, people have sometimes asked me, have you ever seen these types of systems, especially CRISPR systems, in eukaryotic cells? And then recently, Makoto, who is a really brilliant postdoc, he found that there are these CRISPR-like systems in eukaryotic genomes. They are found in either single cellular organisms or even in simple forms of animals. And you can find them in not only the genome, but sometimes even in the chloroplast of some of these photosynthetic organisms. They look very much like a compact version of Cas12. So they have a single nuclease domain, the RUBC domain, and they have a protein rec domain. So it's somewhere in between TMPB and also Cas12. And structurally, it looks just like any other sort of compact form of Cas12. Okay, so anyway, so there's really a lot in nature that is worth exploring. So I usually show this plot that shows how much microbial genomic information has been accumulating. It's really been on an exponential rise. And this trend is growing faster and faster. There's a lot of interesting biology there. Probably all of it is really interesting. And some of it is going to be really useful. And so I think I really encourage all of you to think about if you're trying to solve a problem, chances are for an enzymatic process, nature has already invented it before. So try to look into nature and draw inspiration from there. And you might find something that already will help you solve your problem. Last but not least, so just want to acknowledge my team here at MIT, our collaborators, funding groups, and thank you again. It's really, really exciting to be here today.
谢谢张峰给这么一个非常精彩的讲演啊。这个我们给大家时间问问题，然后最后呢，哎呦，
哇，
easy， 等一会儿，那个我听这个小高我助理说，你妈让你在北大多照几张相，所以待会儿我觉得这是 great 的，那个待会儿在台上你在台上照我们下去
OK
跟大家合张影。
OK，
OK， 你先来。
Thank you, Professor Zhang, for your exciting research in the development of gene delivery system. And I would like to know whether we choose to deliver protein and when we choose to deliver mRNA and I would like to know the comparison of this.
Sure.
Systems.
Yeah, thank you for the question. So I think ideally we want to have delivery systems that can deliver everything, right? From DNA to RNA to proteins, or even small molecules and polysaccharides and all those things. I think the application will depend on what we want to do. For just something like gene editing, you really want to have the gene editor be present for a very short period of time. So that you just edit the DNA and then you want it to disappear after the edit has happened. So there, I think proteins will probably happen the fastest, and then second to that, mRNA would be nice. Delivering DNA, especially long-term expressing DNA, probably less ideal because then you have potential off-target effects. So I think it really depends on what we're trying to do. And it depends on the disease, depends on kind of the modulation we want to achieve. Yeah.
And I have another question. It's about the EPNMA2.
Uh-huh.
It's just a shell in our bodies, and
Right.
it has no the efficiency. It's unlike the virus, like AV and lentivirus, so it can. It cannot escape from the lifestyle or amnosome. So how do you engineer the EPNM2 to
Yeah, good question. So as you know, EPNMA2 or PNMA2 or any of these endogenous gap proteins, they have been evolved over long periods of time. And so I think there are a few different ways to engineer that. You can either do it fully chemically. There are endosomal disrupting reagents. There are drugs that can disrupt endosome. Or small molecules. So you can potentially conjugate or load these small molecules inside of your particle, and they will get delivered at the same time. There are enzymatic processes. There are gamma peptides. There are phospholipases that can chew up lipid. There are other types of membrane disrupting, pore forming proteins that you can potentially combine with this. So I think the idea is simple, but the hard part is really getting it to work. So I think it's a matter of exploring these different strategies in a systematic, serious way, and try to see which one works out the best. Yeah.
Each person asks only one question, okay?
Sure. Yeah.
Thanks for your exciting presentation. I'm really interested in the injection system. As we all know, the spherical shape will have the largest volume. So how about change the shape of the injection system to load more cargo? Like the second system, you say
Sure.
that you edit the plasmid to change the electrical property, to change the injection system.
Yeah. No, that's a great idea. So that's exactly how we're thinking about it too. So if you can change the property of the packaging or the loading, then you can potentially load other things. So that's one way to do it. We can look at the PVC, the photorhabdous syringe system, but there are also other injection systems that you can find now based on that. I'm sorry? Yeah, we have done some of that. And then the other thing that we could do is, if you look at the natural way that some of the other syringe systems work, they sometimes have the payload right on the spike protein. So rather than loading it into the syringe, they just have it on the spike protein. And then when the spring-loaded mechanism pushes the spike across the cell membrane, whatever payload that's attached to the spike gets dragged in as well. So that's another possibility, is you can conjugate not just proteins, but RNA, DNA, whatnot, onto the spike proteins, so that when the spike gets shoved into the cell, whatever conjugated will also get dragged in. So that's another way to approach it. But I think, so that that's learning from what nature has done. So I think as we understand more, we can probably come up with even better strategies.
Okay, there's a question there.
Yeah.
我有个具体的问题，就是我们那个 CRS 那个，我们知道 AAV 是没法重复伪药，因为免疫原因的问题，所以像CRS 或者 VLP 类似的这种，它可以重复伪药吗？然后还有就是我们对它进行了那些改造。
不好意思，那个我中文真是不是特别好，就请你说的稍微慢一点速度。
AAV cannot reduce because of immunogenicity.
Right.
So a system like CECS or other kind of VLP can it reduce?
Yeah, good question. So I think, okay. Okay, so the CS system is probably going to be immunogenic because it's microbial in origin. So the first time might be okay. Second time we may have some problems. The endogenous viral particles, I think, is questionable. They are expressed in the body, and some are at very high levels. So those I think are probably going to be okay. But some are going to be at lower level, or they may be only expressed in the brain and not in the kind of the peripheral systems. So there, I think we'll have to test to see whether or not they're immunogenic. I think just one question. Yeah, sure.
Thanks for your wonderful talk. I'm so interested in the protein PAM2. Yeah, the first story.
Yeah.
Yeah, I was wondering that you just modified the protein so you can carry the mRNA of the TL2. Yeah, you only show data of the TLP. I was wondering that have you ever tried the other protein or genes which actually measure the TLP? I think the measure the protein, it will be harder to be captured.
Yeah, I think that's very insightful. So the PNMA2 particle is about 27 nanometers in diameter. And so the packaging capacity limit is probably 1.5 kb to 2 kb can be comfortably packaged. Up to 3 kb, I think you can get it in. But larger than that becomes quite challenging. But there are other particles that are larger, and I think you can explore some of these other larger particles to get larger, like 5 kb or 8 kb mRNA in. As the volume scales by factor of 3, by power of 3, right? So as you increase the diameter, you can package a lot more things into it. So if you go up to 45 nanometers in diameter, I think you can package quite a bit. Yeah, sure.
I'm sorry, I have to ask this question. I've read the Peking paper at least 3 times. So
Okay.
the really point.
You need to get my information, but it's okay.
Yeah, sure.
So I think you have humanized the gag protein, PEC10, but I think the key point of ALP is the enzyme protein because it's on the outside of the ALP
Sure.
will be neutralized by human antibody.
Right.
So in your paper you mostly use VSVG, it is not humanized, but
Right.
sometimes you use synthetic A in mouse.
Right.
In mouse it's endogenous environment. So in human, have you ever tried any endogenous envelope protein like that?
Yeah, in human, you can try HERV-W, for example, which is another endogenous sort of fusion protein. And I think you can try other ones as well. But I think the challenge with the membrane-bound system is that you need to figure out a way to be able to scale the production. And if you have to use cells to produce them, then it's going to be very challenging. So we are, for the most part, we're shifting away from membrane enveloped VLPs, and we're just using protein-only particles. Yeah.
Okay, good.
Sonny.
Yeah, then you.
I wonder the efficiency of ECIS because I noticed that you didn't show any light field image corresponding to the transfection. And I want to know, and I noticed that there are still many unspecific transfection using ECIS. Because I noticed that there are more or less some just positive cells
Sure.
when you use different pairs of recognition molecules.
Yeah. So I guess which question do you want me to answer? The first one, maybe. Yeah. So the efficiency we haven't done a very thorough characterization of the efficiency yet because it was a little hard to figure out what fraction of the syringe that we have are fully functional syringes versus syringe-looking like things that don't have a fully formed spring-loaded mechanism. So we haven't done that quantification. Although you can quantify based on the amount of Cree protein you're delivering and the activation, but we didn't feel that was as useful. So we haven't done that experiment. But we'll do that as we sort of study it more.
And regarding to the unspecific targeting, what would you do next to figure out?
I think that has to do with the non-specific binding. So I think they're probably finding the right proteins, the right protein target, and also the right antibody is going to solve the problem. Because if you look at AT5 versus the EGFR DARPin, those are fairly specific. And so I think it really has to do with getting something that is specific.
Okay, you asked one question.
Hello, Professor Zhang.
Yeah.
My question is about you mentioned the omega system and
Right.
many other enzymes such as integrase and reverse transcriptase have also shown their potential on genome editing. And some of them are CRISPR-free. And I also noticed your approval on Patrick Schu's work on Twitter. And I'm curious about your perspective on the future application of these non-CRISPR systems. Thank you.
Yeah, thank you for the question. I think it's very, very promising and very bright. Nature has come up with all sorts of things, and retrotransposons of the LTR or the non-LTR variety are very interesting systems for potential RNA-based gene insertion enzymes. The IS-110 transposons that Patrick talked about, I think is also a very interesting class. And there's probably many more. So I think taking a look into transposable element diversity will probably help us find many more tools. But I think it's becoming more and more challenging because the obvious ones have strong signal that probably have all been found. But I think that the promise lies in as we get more data from more special ecological niches where people have not sampled before, there could be very unique systems that have been evolved, and we can find those. Or there may be some very, very low level, low activity, and also low abundance systems that are scattered kind of a little more broadly that we might be able to find. I'm excited that with AlphaFold 3 and the ability to predict interaction between protein and nucleic acid complexes, we can probably set up a more systematic way to mine them. And you need a lot of compute to be able to do that. But I think that will also get solved. So yeah, I think it's an exciting direction. But I think it will require more power and also more kind of analytical sophistication.
Okay, you please.
Yeah.
Thank you, Professor Zhang. I have a general question. For your opinion, what's the future of gene editing technologies, and especially what do you see is the development of nowadays
Yeah. So I think the future of gene editing technology, especially in therapeutic applications, is really limited by delivery technology. We need to have ways to deliver to tissues other than eye, blood, and the liver. There are kidney diseases that we know are caused by single gene mutations. There are muscular disorders. There are even brain disorders that are single gene. And so if we have the ability to deliver to those tissues, I think we already have the gene editing technology to go and treat them. So I think really putting more effort into developing better, more robust delivery systems is also going to take gene editing, make it realize more impact. As for gene insertion, I think what I was saying earlier, looking at transposable elements, I think can potentially provide more substrates for engineering. The thing about transposable elements is that they are selected to be low activity. A host doesn't want highly active transposable element. It's destabilizing for the genome. And so I think we can find a lot of these things, but then we have to engineer them to become more active. And potentially also we'll have to work on specificity. So I think there's work, and I think exploring the diversity is going to make the work potentially go faster. And computational design, de novo protein design might also help us make more progress on that front, or maybe some synthesis of the two. So that's what I think.
Great. Yeah.
Thank you for the talk.
Okay.
For the first part, I think other groups like Michael Eloise has developed similar vehicle by viral-like protein. I don't know which paper is published earlier. So I wonder what are the advantages of your system? How we compare them parallel? Thank you.
Yeah, great question. So I think the virus-like particle system as a general approach, I think is very interesting. And it's potentially more engineerable because there are biological components. It's easier to engineer proteins than to engineer lipids, for example. So I think that's the thing. We have not done comparison because we realize that the PEG-10 system that we've been working on, we need to optimize it more for production, for scalability, before it's even worth making a comparison with other systems. The advantage, I think, really lies in these protein-only systems. And especially having a system that's made of endogenous proteins could be immunologically more tolerated. And that could make redosing and application safer. Yeah.
OK， 最后一个问题。
I have a
Okay.
question about the
Very
ECIS. You mentioned that you truncate the ECIS and replace it with HHMI. My question is, how do you select this binding domain, and how do you make sure that it can inject the payload into the cell rather than just bind it to the surface of the membrane?
Yeah, good question. So we find these surface markers based on what people have sort of discovered from past. So it's based on knowledge from other people's work. And then we try to identify antibodies or binding domains that have been shown to work really well for that target. So if you wanted to target hematopoietic stem cells, there are CD117 targeting antibodies reported by several labs now, actually, that we can use and target those cells. So that's how we go about it. We haven't been focused on engineering these binding domains. It's more how do you couple the binding domain to the system effectively. Specificity-wise, I think it's really doing the control experiments to make sure that a system that doesn't have the spring-loaded injection mechanism, or it doesn't have the spike protein or whatnot, that can still bind to cells, don't have the signal. And that can show that binding alone or even endocytosis doesn't lead to functional activity from the injected protein payload. So that's how we show that they have to be injected rather than just binding to the cell to get activity. Sure.
我知道大家还有很多问题，
Okay.
但是12点我必须把他送到隔壁。
OK.
因为这是你第一次来北大，所以我们给你
OK.
来这儿合个影。
OK. 好，谢谢。Okay.